stem cell. In July of this year, General Electric, multi-billion-dollar worldwide company, signed an agreement with this company, Geron, to use embryonic stem cells and their derivatives, that is the specialized cells we can make from them, to develop cells in order to screen drugs. So ways to take the embryonic stem cells, to specialize them into heart cells or brain cells, and then use them for pharmaceutical companies to screen against their experimental medicines, or to see whether or not they're efficacious, whether or not they have any toxicity, using actual human cells, not animal models, not cell lines, actual human cells. Finally, iPS cells. This technology has really only been around since 2007, when it was demonstrated in animals first, and late 07 or early 08 when we were able to make these cells from human tissue. They've been very useful at giving us as scientists insight into certain diseases by developing disease-specific iPS cells. So taking samples of cells from people with Parkinson's, diabetes, ALS, creating the iPS cells, and using them to study the disease very early in its course. However, there are some caveats that need to be mentioned with respect to iPS cells that at this point in time we're still trying to figure out whether or not they're ultimately going to be useful clinically. For example, in a recent study that was just released this week, the cell that was taken from the patient, in this case let's say a skin cell, and transformed into an iPS, still has some genetic signature of the skin cell. Whether or not that affects the ability of the iPS cell to function like a stem cell, we don't know. But it's certainly something scientists are investigating. Additionally, iPS cells are not genetically to embryonic stem cells. They function like them in a lot of ways. But we know that depending on how you make the iPS cells, there can be anywhere from 0.1 to 1% of the genes different between the two. So there are roughly 30,000 genes in the human genome. And we can survey the expression of those genes. And when we compare embryonic stem cells to iPS cells, anywhere from 0.1 to 1% of the genes are different between the two. Is that meaningful? We don't know yet. That's the point. We don't know yet. Additionally, iPS cells may, in some cases, be more tumorigenic than embryonic stem cells. So when we specialize embryonic stem cells, we have to be very careful not to leave any of the unspecialized cells around. Otherwise, they have the potential to generate tumors. We know that from experiments in animal models. iPS cells, in some types of derivation, have the potential to be more tumorigenic. Again, something that's being worked on, which makes them exciting scientifically at this point. Maybe one day they'll be useful clinically. They have lots of upsides, but it's a work in progress. And so I just want to conclude to give you a little bit of insight on what's going on at the University of Michigan. Since you're all here, because you are U of M alums, we are at the Center for Stem Cell Biology pursuing research with all types of stem cells. We have investigators who do adult stem cell research, embryonic stem cell research, and study induced pluripotent stem cells. We are continuing to expand the faculty for the center. And currently, for a particular project we're working on, we now contain, or now have, one of the largest collections of human embryonic stem cells. Of course, I should say that those stem cells were derived in other states, but we have them now in our laboratory to work on a particularly interesting project that I think really has a chance to make a big impact, not only in medicine, but the field of stem cell biology, and that is to work on mapping the ethnic diversity of existing human embryonic stem cell lines. So to this point, estimates have about 700 or so lines of embryonic stem cells out in the world. But what we don't know is what is the ethnic diversity of those lines? Where do they come from? And which populations are represented? Which populations are under or not represented? And that's important for two particular reasons. One is that if you want to study diseases that target or affect specific backgrounds preferentially, you need to know, you need to have the right genetic background to study that disease. So by surveying the existing lines of embryonic stem cells, we will then know which lines or backgrounds are represented and which aren't. Additionally, if these lines and embryonic stem cells are going to be used to screen new compounds, for example, new experimental medicines, it's important that the companies know that their medicines affect positively or negatively the population that they want to target, or are not specific to just one population, that they globally work across different genetic backgrounds. There's a whole field of pharmacogenetics where small mutations in your DNA affect how you respond to certain medications, what side effects you have, and additionally what diseases you're susceptible to. So this is really a nice niche that the university is focused on right away and has a chance to make a unique impact in from the very beginning. And then there's the A. Alfred Taubman Medical Research Institute Consortium for Stem Cell Therapies. That's the one that formed post-Proposal 2. And they are now in the process of staffing that, equipping that facility in order to derive new lines. But the most fascinating part of that is it's been 9 months since they've started that process, and they are still now getting clearance from the regulatory agencies to begin that work. And it's been 9 months up to this point. Just another example of the regulation and control over this type of research and the way the university is following that research, or following those guidelines in its research on stem cells. And I'd just like to conclude by saying, you know, as a scientist, for us, it's really a race against treating or curing these diseases. And for us to have the best chance of winning that race, we really need all the weapons at our disposal: adult stem cells, embryonic stem cells, and iPS stem cells. And the university is fully committed to pursuing those types of research, that aggressively, and pursuing that type of research. So with that, I'll take any questions. Yes?
I just wondered if you could comment on somatic cell nuclear transfer and whether that's something that...
That's illegal in Michigan.
Is it illegal?
It's illegal in Michigan, so it's really a non-issue. Oh, sorry, somatic cell nuclear transfer. She said, can you comment on that? And my comment was, it's illegal in Michigan. We don't do it.
Is it something that you think is important, though, for the regenerative medicine to proceed?
Well, there are other ways to obtain those iPS cells, for example, obtain patient-specific stem cells. If we're able to overcome the barriers that exist now with iPS cells, those may be a way, a substitute. Yes, ma'am.
Well, first of all, I'm Senator Liz Brader. I wanted to say how proud I am of everything that's going on at the University of Michigan. Sorry some of us were late because we were in session, but welcome. And I had a question going back to the process of using the iPS cells. You said you injected a gene to specialize it, but what's the source of the genes?
Human genes. They have, you can go to it's a kind of like a DNA repository and select 1 or 4 of these genes, and they'll send you the sequence. It's human.
So they're just harvested from people?
Oh, the source
How do you physically
of
get them?
I don't know where that particular, I don't know what type of cell it came from. It's human DNA, and all of your cells, your skin cells, contain the same set of DNA. I don't particularly know which cell type it was harvested from.
But that part of it has never been controversial in terms of all the political ramifications that we are aware of with stem cells. That part has never been challenged.
There's no identification, there's no way you can link a particular person to that DNA that you use to put into the cells to engineer iPS cells.
And that's coming from adult stem cells, or I mean adult cells.
Yes. Well, I don't know the exact source. It makes sense that it's probably from an adult skin cell, but I don't know that for sure. Did you want to add something?
So for purposes, because it's being recorded, so the questions are about the